Collect the 5 nanoparticle tubes from the refrigerated Proteograph Assay Kit box. Do not uncap the nanoparticle tubes until immediately prior to loading. Do not store the nanoparticle tubes on ice. Centrifuge the samples. Carefully pipette at least 250 microliters of each sample into a sample tube. Minimize aspirating residues, which may settle at the bottom of the tube. Minimize bubbles forming at the surface of the samples. Keep the samples refrigerated or in ice until ready to use. Place the sample tubes and control tubes in tube carrier 1. 16 samples and 2 controls must be loaded for the method to proceed. Ensure that the tubes are uncapped prior to loading. Ensure that all sample and control tubes are fully seated into tube carrier 1. Uncap the nanoparticle tubes and place in tube carrier 2. Ensure that the nanoparticle tubes are uncapped prior to loading. Ensure that all nanoparticle tubes are fully seated into tube carrier 2. Ensure all sample, nanoparticle, and control tubes on both carriers are uncapped and barcode labels are visible before proceeding. When the final labware check screen appears, confirm proper loading of the SP100 deck.